Welcome students. Today's topic is microorganisms. Just look around you. Can you see all the things around you? No, you cannot see all the things around you. There are organisms which are very small and which are not visible to your eyes. These organisms are called as microorganisms. They are visible only under the microscope. The other name for microorganisms is microbes, and the study of microorganisms is microbiology. Now, where are these microorganisms found? Yes, they are found in air, water and soil, and not only that, they are also found within our own body. Are they active all the times? No, they are active only under favorable conditions, and they remain inactive under unfavorable conditions. And these microorganisms are basically classified into 5 groups. The first one is virus. Let us first learn about virus, and the second one will be bacteria, third one algae, fourth one fungi, and finally protozoa. First let us learn about virus. The name virus itself means poison in Latin. The study of virus is called as virology. And this virus is an intermediate between living and non-living beings. And when you take a virus, it is very small. When you compare it with bacteria, it is smaller than bacteria. That means how many times smaller? It is 10,000 times smaller than bacteria. And virus cause many diseases not only in plants and animals but also in human beings. Now let us study about the cell structure of virus. First of all, virus has the core, DNA or RNA. Then what else it has? Surrounding this core, DNA or RNA, it has a protein coat. In some virus, surrounding this protein coat, an envelope is also present, and this envelope has numerous spicules in it. This spicules helps the virus in getting itself attached to the host. Next, we are also telling that virus is an intermediate between living and non-living. So, it exhibits both living characters and non-living characters. Let us see what are the living characters it is exhibiting. The first living character it is exhibiting is, it is responding to stimuli. It is responding to stimuli. Not only that, it responds even to heat and chemicals. And the third point will be, it reproduces within the body of the host and produce the copies of itself. Next, let us learn about non-living characters, and the first character will be no cytoplasm. And the second character will be, that is, it is active only within the living medium, and outside the living medium it remains inactive. So, inactive outside the living medium. And the third point, it could be crystallized. These 3 points indicate that virus are also non-living, and it also exhibits living characters and non-living characters. And in virus, we have numerous shapes in it. That's all about virus. Next, we'll move on to bacteria. Yes, bacteria is unicellular. That means it is single celled. And very characteristic feature is, it doesn't have any nucleus. And these bacteria are also very small. That means 1 micrometer, or you can also represent it by μm, 1 micrometer to 5 micrometer. This will be the size of the bacteria. Now you can guess how small the bacteria is, and the study of bacteria is called as bacteriology. And depending upon the modes of respiration, bacteria is classified into two types: aerobic bacteria and anaerobic bacteria. Now what is this aerobic bacteria? Those bacteria which can survive only with the help of oxygen are called as aerobic bacteria, and those bacteria which can survive without oxygen, they are called as anaerobic bacteria. Next we will move on to the cell structure. Before going into cell structure, we must also know that the bacteria is the first formed living organism on earth. Now we will move on to the cell structure. Okay, we already know that the bacteria has a characteristic cell wall. It has a characteristic cell wall in which you can see cytoplasm being present. This is a cell wall, and you can see cytoplasm present. You don't have a characteristic nucleus is absent, or nuclear membrane is also absent, but you have DNA, circular DNA present in the middle, and you also have ribosomes are present here. And what are these ribosomes doing? These ribosomes are doing the function of protein synthesis. And you have flagella here. What is the function of the flagella? Yes, flagella helps in locomotion. That's all about the parts of the bacteria. And next we will move on to how this bacteria is classified based on their shapes. And whatever the other important organelles which are absent in bacteria is mitochondria is absent, Golgi bodies, those things are absent in bacteria. And depending upon the shape of the bacteria, it is classified into four types, and the first one will be bacilli, second one will be cocci, third one vibrio, and fourth one spirilla. And the first one, bacillus. Bacillus, plural is bacilli, and this is rod shaped. Rod shaped. And the second one, cocci, is spherical. And third one, vibrio, is somewhat comma shaped. And the next one, spirillum, is spiral shaped. These are the four types of bacteria depending upon the shape of the bacteria. Next we will move on to bacteria depending upon the flagella. How much flagella is there? How this flagella is arranged? According to that, bacteria is classified into 5 different types. And the first one will be monotrichous, second one lophotrichous, third one amphitrichous, fourth one peritrichous, and the last one atrichous. Okay, in monotrichous, this is our bacteria. One flagella will be found at one end. This is called as monotrichous. Lophotrichous will be tuft of flagella, that is many flagella will be found at one end. Amphitrichous will be tuft of flagella at both ends. Peritrichous will be flagella throughout. And atrichous will be no flagella. So these are the 5 types of bacteria classified on the basis of the number of flagella and the arrangement of flagella. Again, we also have two other types of bacteria, that is photosynthetic bacteria and chemosynthetic bacteria. This photosynthetic bacteria, they can prepare their own food. Those bacteria are called as photosynthetic bacteria, and chemosynthetic bacteria are those bacteria which can prepare the food only with the help of chemicals. And reproduction here by means of binary fission. That's all about bacteria. Next we will move on to fungi. Okay, fungi is mainly found in dark places. Mainly found in dark places, and it lacks chlorophyll. Next one will be study of fungi is called as mycology. And the last one, here you can see unicellular and multicellular fungi. Both types of fungi could be seen. Let us first see about unicellular fungi. Here you can take unicellular fungi, example you can take yeast. Let us see what is the characteristic feature of the yeast. Yeast grows in all medium whichever contains sugar. In all that medium which contains sugar, yeast could be seen growing. And the shape, if you take, it is almost ovoid in shape, and you can see cytoplasm will be there, and outermost is cell wall. Cytoplasm is seen. Characteristic nucleus is also seen, and vacuoles are also present. And three more important points in this unicellular yeast is, the first one, it helps in fermentation. Why it helps in fermentation? Because it is anaerobic. That means it can survive without oxygen. So it helps in fermentation. And next, when we are talking about the reproduction, here reproduction is mainly by budding. Next we will see multicellular. Multicellular fungi, here we will take agaricus as the example. When you are taking agaricus, there is a, when we draw the shape, it is somewhat like this. Here you have an umbrella shaped body. This umbrella shaped body is called as fruiting body. Fruiting body. This body contains many slits in it, and these slits are called as gills. And what is specific about these gills is, these gills are so important in which spores are present, and these spores in turn help in reproduction. And then you can see below, under this fruiting body, you can see mycelium present. What is this mycelium? These mycelium, in turn it is made up of hyphae. Singular is hypha and the plural is hyphae. What is this hyphae? Thread-like structures which are present in the mycelium are called as hyphae. And what is the function of this hyphae? The function of the hyphae is to carry on all the nutrients from one place to the other place. And here reproduction is mainly by means of fragmentation and spore formation. Next we will move on to algae. Where is algae found? Algae is mainly found in moist places. Let us see where is algae found. Algae is found in moist places. And second one, algae is very rich in chloroplast, or very rich in chlorophyll pigments. So since it contains a lot of chloroplast in it, it can prepare its own food. And what is the study of algae called? Study of algae is called as algology. And here also you have unicellular algae and multicellular algae present. In this particular lesson, we are going to learn only about the unicellular algae. Here, you are going to learn about unicellular algae Chlamydomonas. This Chlamydomonas is very simple. It is unicellular. Let us see what is the shape of the Chlamydomonas. It is almost oval in shape. And here you have two flagellum coming out like this. And the very characteristic feature of this algae Chlamydomonas is the cup-shaped chloroplast. Can you see this cup-shaped chloroplast? Yes, very identical and it is very characteristic, and this is your cup-shaped chloroplast. Outermost will be the cell wall. We have a characteristic nucleus, and this nucleus is located within the cup-shaped chloroplast. Yes, and the cytoplasm is also seen. It is seen between the cell wall and the cup-shaped chloroplast. We have another body which we call it as pyrenoid. Pyrenoid is also present. And we have two flagella which helps in locomotion. At the base of the flagella, you can see two vacuoles present, which are contractile vacuoles. Again, a dark color red spot is present, which we call it as eye spot. That's all about the parts of the Chlamydomonas. Then we shall see about the reproduction of Chlamydomonas. In Chlamydomonas, the reproduction is by sexual and asexual method. That's all for today's class. Thank you.